pick up on this topic of mutations. So, what happens when some sort of change or alteration occurs to our DNA, and how does that end up resulting in a change in everything that comes from that? So, when we talk about mutations, by definition, it's usually some sort of mistake that has occurred, some sort of change, some sort of alteration that has happened while DNA is replicating in our cells. So that change typically occurs by inserting an incorrect base, or those mutations can also happen when we talk about going through processes like crossing over, where we talk about big chromosomal mutations as well. We're going to focus a little more on the point mutations here, although I'll do a quick refresher on the chromosomal mutations because we focused more on those chromosomal mutations when we talked about mitosis and meiosis already this year. Regardless of how that mistake is happening, you've got a lot of enzymes whose job it is to recognize those mistakes, fix them, or shut down the cell before it can go through mitosis or meiosis with that mistake in place. Are those enzymes and those self-correction mechanisms perfect? No, and of course things slip through. But DNA polymerase is just one example of an enzyme whose job it is to not only produce those new DNA molecules, but then also go back, check for mistakes, and hopefully fix them. So here's the thing, how do we get mutations in the first place? Well, for the most part, those mutations are spontaneous, which really kind of just means that we don't know why they happen. For most organisms, there is sort of a low-level background mutation rate that occurs just naturally. You go through enough rounds of cell division and you're bound to make a mistake, and this is sort of the frequency at which it happens based on the DNA replication mechanisms that occur in that particular organism. On the other hand, there are definitely things that are in our environment that we know increase that rate of mutation, and these are things that we call mutagens. So a mutagen is any sort of agent, things that are chemical, UV light, radioactive elements. Regardless, they will increase or induce the frequency of mutations in an organism. So when we expose an organism to a mutagen, we see that rate of mutation increase above what that typical background mutation rate is. So if we would expect a fruit fly, for example, to create a mutation once every 1,000 times it goes through DNA replication, we introduce a mutagen, and now we may see that rate jump to 1 in every 500 times it goes through mitosis or meiosis, goes through that DNA replication. So we're clearly causing more mutations to occur because that mutagen is around. So if you think about it, a lot of mutagens that you know are actually cancer causing. Chemicals in your cigarettes, exposure to sun without using your sunblock, that kind of stuff. That being said, when we talk about those types of mutagens, we may more specifically call them carcinogens. So mutagen's a bigger category of any mutation. If it specifically causes cancer, then it's a more specific type of mutagen called a carcinogen. But regardless, those mutagens are things that increase the rate of mutation in an organism. So here's the next thing. When I mention mutations to most students, usually the first thing that comes to mind is that that mutation is going to be damaging to the cell. And oftentimes they're correct, but take this all the way back to natural selection and evolution. When we look at mutations, we really have three big options as to what that mutation ends up doing to the cell itself, and as a result, the organism as a byproduct. So naturally, most of those mutations, by changing the protein cause the protein in the end to be malfunctioning, and so it's deleterious or harmful to the organism to hang on to that mutation, which means that that mutation will probably get weeded out of the population relatively quickly because those organisms are not surviving and reproducing as well as someone without that mutation. That being said, mutations do have the potential and have had beneficial effects to organisms. If that mutation somehow occurs and produces a protein that actually functions better than the current version of the protein in the population, then that organism may find a benefit in increasing its survival and reproductive rate, so the mutation eventually over time becomes more and more common if it helps them be better suited to their current environment. And then there are plenty of mutations that actually have little to no effect at all. You're all sitting out there right now with mutations in your DNA, and yet you have no idea without actually running a DNA sequence on yourself. And that's because they really aren't affecting the way that protein gets produced in the long run. And so your proteins function just fine, and so natural selection doesn't really care, and we don't notice just looking at you or even understanding the way that you function because the protein is in fact doing its job. So we're going to focus more on this idea of point mutations, and specifically we'll start off with substitutions. So a substitution mutation is when I take a single base and I replace it with something else. So perhaps I was going through the DNA sequence, it was supposed to be an adenine that got added next, and by mistake a guanine got replaced. So that's what we're talking about, swapping one for another. And here's where I can take a look at having three potential outcomes as a result of swapping one base for another. If I end up in a situation where I change the codon and now I code for a different amino acid, now I'm talking about a missense mutation. And this often is more likely to occur if I change the first base in the codon as opposed to the last base in a codon. Because if you remember back in your codon chart, there are way more codes for your amino acids than there are amino acids. So 20 amino acids would correspond to 20 codes, but there's actually 64. Now, 4 of them are corresponding to stops and starts, but the other 60 are considerably more than the 20 that you would need. So there are definitely amino acids that are being coded for by multiple codons. So if I change the first base, that's more likely to change the amino acid than if I change the last one. So I'm going to jump over the nonsense for a second and go down to the silent mutations, because these mutations are the ones that I was just alluding to a moment ago, where most of us are probably sitting around with mutations and yet not really experiencing any difference. And that's because that substitution probably occurred in that third base of the codon. And it's silent. It doesn't end up changing what the amino acid is at all. And that's what this idea of this wobble effect is. You've got more than one codon, so there's redundancy in our genetic code, which is probably a good thing from an evolutionary standpoint, because mistakes are bound to happen. So if we have a way to not alter the final product, even though we've made a slight mistake in the way that we got there, that's probably a good thing. So missense still codes for an amino acid, just the wrong one. Silent mutations code for the same amino acid, even though I've changed a base. And then occasionally you end up with these nonsense mutations. So there are 3 stop codons that exist in our genetic code. And if in the process I take a codon that should be coding for an amino acid and instead turn it into a stop codon, this is a nonsense mutation because now I've stopped my protein short. So what kind of effects can these guys have on the protein itself? Well, silent mutations you're not going to know, right? Still going to code for the same amino acid, so as far as the protein is concerned, nothing has changed. The missense and the nonsense mutations, the amount of potential harm or potential change they can create really depends on where that amino acid is being changed in the entire protein's structure. I would say that More often, the nonsense mutations tend to be problematic. However, if I've got 10 codons in my protein and that stop codon got changed instead of the 10th codon, it's now 9 codons, maybe that doesn't have as big of a problem. However, if it should have been 10 and it's now only 3, clearly that's going to be a protein that doesn't do its job. A missense mutation, once again, depends. Where does it happen? If it happens in something like a bend in the active site because this is an enzyme, then yeah, probably has a pretty big impact. That being said, if it's not necessarily in a place that is very active in what it's doing and it's more just kind of being part of the overall structure, then maybe it depends on what kind of amino acid it changed for. Amino acids have chemical properties just like every other molecule. So we have amino acids that like water and are hydrophilic. If we have amino acids that are hydrophobic and don't like water. So if I happen to swap out an amino acid that was hydrophobic for another hydrophobic amino acid, it's probably not going to have a huge effect on the way that that protein folds, and so the protein probably does its job just fine. That being said, if I take an amino acid that was hydrophobic and now swap it for one that's hydrophilic, that probably is going to change the way the protein folds, and as a result, might have a bigger effect on the way that that protein works. So, as far as those nonsense and missense mutations go, the effect they ultimately have on the protein, you really kind of have to know more information diving into a specific type of mutation. So here are just some six examples of substitutions. This one's just really just showing you the substitution itself. So my normal gene should have had an A, and now I swapped in a C by accident, and this didn't get caught by our typical DNA polymerase proofreading. Here's another one. So should have been a G, now it's a C. I changed the amino acid, and so there's an example of a missense. Here I should have had a G, but it got swapped for an A, and as a result, I now have a nonsense. It's too short. So it should have been, let's see, 1, 2, 3, 4, 5, 6, 7, 8 amino acids long, and now I've only got 4 of them, so it's cut it literally in half. So let's talk about once more that idea of what's the effect, right? A lot of times these point mutations aren't overly harmful, but they can be. Sickle cell anemia, for example, is an example of a point mutation, a substitution mutation that actually has a really big effect. It's a single base swap, and what it ends up doing is taking your typical red blood cells, which should look more like this donut shape here. So they're bendy and flexible and can squeeze through really tight capillaries and things like that to carry oxygen from your lungs to your tissues. Instead, that point mutation causes parts of that hemoglobin protein to bend and misshape, and so it ends up creating red blood cells that look like this. That hemoglobin is the major component of your red blood cells, so by causing that protein to misshape, misfold, you end up with this sickled cell looking red blood cells, and these guys tend to be hard and sticky, and they've got harsh ends to them, so they'll get stuck in capillary beds. They do particular damage to a patient's liver, especially if this isn't managed and treated properly, plus that they're not getting the oxygen delivery system functioning the way that it should. So their tissues aren't getting the typical oxygen levels, so they tend to be run down, fatigued, or easier, run out of breath easier, in the sense that they're not able to bring in enough oxygen to their lungs, but their body isn't good enough at getting that oxygen from their lungs then to the tissues. And all it was a single base substitution. One letter got swapped for another, one amino acid got swapped for another, and this one actually does have a pretty big effect on the way that it functions. So while we're still talking about swapping in letters, We can also talk about frame shift mutations, and this one isn't like the substitution where I'm taking one letter and exchanging it for another. Instead, I'm either inserting something extra or I'm deleting something. And anytime we insert or delete delete letters that aren't in multiples of 3, every codon beyond that point of insertion or deletion now gets read incorrectly. We talk about it shifting the reading frame. Because remember that I have to read my bases 3 letters at a time, 3 bases at a time. It's a triplet code, it's a codon code. So insertions would be the addition of one or more nucleotides in the sequence, deletions would be the removal of one or more. And if I don't do it 3 in a shot, I'm going to shift everything from that mutation point forward. And these almost always end up changing the resulting protein because I'm changing everything downstream or past that mutation. So Crohn's disease, we believe, is an example of a frameshift mutation. And as a result, they've got a lot of issues with their ability to digest certain materials because they're not able to produce the enzymes that they need in order to do that. So I kind of like to think of this in a host of ways. I think the sentence analogy is probably one of the best ones. So here's your typical sort of first grader sentence. Everything is in groups of 3, and so long as everything's there, it makes sense. But if I were to remove a letter, I now have to shift everything so that I've got 3 bases at a time, and now clearly that sentence does not make sense at all. All right. Now, we can have mutations that are just these point mutations, single base swaps, right? But remember, we also talked back in mitosis and meiosis about the fact that I can have changes to my entire chromosome structure, in which case I'm talking larger scale potential mutations and issues as a result of that. And these often are associated with mistakes that occur during crossing over. So, one potential option for those chromosomal mutations is a chromosomal inversion. So, when we go through DNA replication, and we go through that crossing over process during meiosis, we're supposed to take the genetic information, same pieces, and swap them correctly with the corresponding homologous chromosome. But sometimes in the process of doing that crossing over, the chromosome that is being moved actually flips. And so as a result, what I should have had was a sequence that looked like this. I've now got this piece of information that's completely backwards. And we believe hemophilia is actually a potential type of chromosomal inversion that occurred. So hemophilia is a bleeding disorder. Depending on how severe that type of hemophilia might be, something even as small as a paper cut can just bleed and bleed and bleed and bleed and bleed, and you're not making the clotting factors that stop that bleeding from occurring. Here's just another example if you like sort of pictures. So here's what should have been the correct order of the alphabet. The inversion happened, and now I've got this middle section here reversed. We can also have gene duplications. So again, fault of crossing over. So it should be swapping the same amount of information for the same amount of information, but it's not always very good at that. So I end up as a result having this particular chromatid that's going to be a little too short in the end, and this one that's going to be too long. So if I have the gene for eye color here, this one may now be missing that gene for eye color, where now this one has 2 copies of it. Translocation, right? Once again, probably a fault of crossing over. When we go through crossing over, I should be matching up homologous chromosomes. So chromosome 1 to chromosome 1, chromosome 17 to chromosome 17. And yet something occurred during that synapse process that caused chromosome 1 to now merge with chromosome 17 instead of 1 and 1, 17 and 17. And so when it goes to that crossing over process, I'm now swapping information with the wrong set of chromosomes. So it's kind of like taking a book in the library from the nonfiction section on history of World War II, and I'm moving that World War II book into the middle of the Shakespeare section. All right, now somebody goes looking for that book and has no idea where it is because it's not in its proper location. And your DNA runs into those issues here as well because they're going to look for certain information expecting it to be one place. It's not, and they don't know where to start looking for it. So again, there's a whole complex system of enzymes, DNA polymerase just being one piece of that, that are active during your S and G2 phases of interphase of your cell cycle. And the whole idea here is to catch these DNA mistakes, catch these mutations, fix those mutations so that that cell doesn't go through mitosis, allowing that mutation to persist. Does it always do its job? No, of course not. Certainly mutations make their way through. But more often than not, it does a pretty good job at doing what it's supposed to and fixing the problem. And if it can't fix the problem, then it's supposed to cause your cells to self-destruct, initiate apoptosis, and destroy the cell. Once again, does it always initiate that system? No. Do those mutations get through? Of course they do. But for the most part, our cells are pretty good at fixing the problem ahead of time.